Hello, good afternoon. Hello. Hello. Hello.
Now the microphone is on now. Can you maybe check if we can hear better?
I just checked.
Yeah.
Because you're also parallel streaming
Yeah.
on Google and so.
এখন আসছে তো? Okay.
Better?
Yeah, better.
Thank
you.
So in our lab, we really work with all the representatives from these land plant groups. We now grow hornworts, liverworts, mosses, not the lycophytes, but we have a genetic system within the ferns, and of course we work with angiosperms, not a problem there. Now when you look at this phylogeny here, you may wonder what came before. And the Cygnumataceae, this is actually a group that was only put very recently as the sister group to all land plants. The Cygnumataceae, they are like thread-like algae. They look really, really simple, not complicated at all. They don't grow three-dimensionally, but they really are proven now by molecular markers to be the sister group of all land plants. So it's not the charophytes, as this drawing here may suggest, but it's this thread-like algae as the sisters. So when you consider all the land plants together with their most closely related group, the Cygnumataceae, you see already that there's quite a lot of things that they have, right? So these guys here, they are able to communicate via ROS signaling. Some phytohormone signaling already is present in those, and they can grow in an apical way. And then the land plants really, they have evolved the haploid lifestyle. You know, the gametophytic, sporophytic lifestyle, and they all have a multicellular sporophyte, which becomes really important later on in plant evolution. They have antheridia and epigonia. They already have evolved like dispersal structures that can be dispersed by wind, the so-called spores with an exene, and embryogenesis. This is really an important innovation because it allows the plants to have a stage where they are not vulnerable to desiccation. And as soon as a plant you come to land, of course you are exposed to desiccation, severe drought, severe heat, severe UV radiation. And this little structure of an embryo really helps to endure those periods of unfavorable conditions. Now this is what all the bryophytes can do, and of course the tracheophytes as well. But then the most recent common ancestor of all tracheophytes had a free-living sporophyte that was independent of the gametophyte, and that was a real novelty. Because that sporophyte, of course, is diploid. Now, diploid organisms can do the cooler things because they have a backup system, right? When you have a mutation in one of the alleles, there's another allele that backs up the wrong mutation. So this was quite a major step. And then we have the ferns branching off, and here's where there's another very important step happening at the base of the seed plants. So seed plants are really rocking the earth, the planet Earth, right? So basically everything we eat, the way we exist, strongly and heavily depends on seed plants. No plants, no life on the planet. If the plants aren't doing well, we will be hungry. And they are so successful because they have this novel thing which is so normal to us called seeds. So they have dispersal units that can be dispersed by, say, wind, or they can be dispersed by animals, which also which embed the embryo, this very durable stage, and they have a mean of transporting these propagative entities for long distances. So range expansion is easier with seeds. The same as later on when the angiosperms have evolved carpels. But here at this step, Also immobile sperm occurs. So the plants we had before, they all require water for reproduction. Yeah, so their sperm cells, they have flagella. And if they don't get any water, they can't swim to the egg cell for fertilization. So that's a major inhibition. Because when it's dry, you can't propagate. That's a bummer. And they have also invented the micropyle. So you have the gametophyte, and that is covered by covering sort of tissue. So it's protected, doesn't dry out so much. And then the angiosperms, of course, our favorite ones, right? So they have carpels, and those carpels develop into fruits. And the carpels is basically what I'm going to talk about later on. And also double fertilization. And double fertilization, of course, is closely connected in evolution with the innovation of carpels. Now, as beautiful as these wonderful steps in the or these accumulations of innovations during land-plant reproduction may sound, they're not so easy really. Because they come at a cost. Now this doesn't move. Here we go. Okay, that comes at a cost. For example, when you invent an embryo that is drought tolerant, it needs also a network. Oops. Oh, what did I do?
Right hand side.
Okay. Yeah. Because you will need networks, genetic networks that recognizes drought and prepare the embryo for almost desiccation state. So while embryogenesis is an innovation, the innovation of networks for drought resistance are another innovation. It's just that we need to think about these innovations also in terms of what kind of molecular mechanisms did those innovations really require? Not just the organ that is new, but also what's really behind developing a new organ. If you have an immobile sperm, you need to come up with a new mean of transport, right? If it isn't water, it's got to be air, it's got to be animals. If you have seeds, all of a sudden, the sperm cells cannot enter the egg cell anywhere, right? They are surrounded and protected, so you need to come up with a hole somewhere in this protective code. And you need a sort of signaling pathway, so the egg cell and the sperm cells, they find each other. Another signaling pathway. Directed pollen tube growth. And then when you have double fertilization, it all sounds really nice. It matures at the endosperm and the embryo, they all develop together and nothing is wasted. But this coordination of embryo and endosperm development requires a really tight network. And this is really hard to disentangle. And I'm really happy we don't work with it because it's very complicated. And then when you think of this wonderful innovation of the carpel, you have trouble envisioning that there needs to be long-range signaling so that the pollen tubes really know where to grow. They cover sometimes many centimeters. If you, for example, think of a gynecium of a lily, that's like that big. And the top pollen grain is just that big, right? So this little thing needs to find a way to grow through massive amounts of maternal tissue. This maternal plant is a different individual. So this is a novel process, right? So one individual is allowed to grow through another individual. And that requires, of course, very extensive signaling. And we know that some of the signaling is also related to defense mechanisms, because it would be sort of not a very good idea if the maternal plant would see this pollen tube that's growing through her tissue as an enemy, right? As a phytopathogen. So it's quite a big step that we are going to make here. So while we have innovations in terms of novel organs, novel structures that do require genetic networks to be realized, in the background we also have to solve all these coordination problems of things growing through other things, finding stuff in structures that are becoming more and more covered. So protection always comes at a cost. And this is exactly what we work on in this research unit that you kindly introduced. So in this research unit, we basically focus with a couple of friends in Germany. So we are around 8 research groups. We focus on some of those topics here and try to elucidate on a molecular level how specific organs have originated, and how the genetic networks that are required for signaling, say, for pollen tube growth, for double fertilization, how they came into place, where the parts of those, the genetic parts of the signaling components come from, and how they were rewired. So this is basically what we do. Okay. And as a single group, we mainly focus on carpel evolution. Or well, this is what I'm going to talk today to you about. So why do we focus on carpels? There are 2 aspects to carpels. First of all, most of our food, directly or indirectly, comes from carpels. Everything you eat in terms of grains, fruits, nuts, veggies, maybe not spinach and salad, but all those things that are high in protein, high in carbohydrates, they are derived from grains and from carpels individually. But besides that, on the botany side of things, they really are important because they're considered to be the most important single trait that is unique to angiosperms. Yeah, so that's what we call an odd apomorphy. They are the characteristics of angiosperms. That's where they come, where the name comes from. Because angiosperms means angio means covered in Greek. The molecular genetics of this organ is very well studied. So we know many transcription factors, and we know many phytohormone regulatory steps that are involved in carpel development. Carpels provide a mechanical inbreeding barrier, so many of the mechanisms for self-incompatibility are located on or in the carpel, and of course they are female reproductive organs, you know all that. So we really like them for many reasons. The problem is that they are the most complex plant organ that plants have ever produced. They come with many different tissues, and they all require a large set of transcription factors to work together. So here, just very briefly, this is how a Arabidopsis thaliana gynecium, which is made up of 2 carpels, really develops. It's a tube-like structure that fuses in the end by postgenital fusion. In the inside, the ovules are formed, and you have a transmitting trachea in the center. And when you only consider this region here, which is the most complicated one, in the year 2013, we knew already that there were 86 transcription factors required to work together to form this region here. This is the carpel margin development, which then will give rise to the ovules. And that's what I mean by developmental complexity. Many different tissues, lots of individual transcription factors from different transcription factor families that coordinate its development. Now, when you think of the ancestral gynecium, if you would like to understand where the gynecium comes from, we need to look at the ancestral gynecium. This didn't look so very complicated. So, for example, It was also tube-shaped. It wasn't closed by postgenital fusion, but just by some slime secretion. There were only a few ovules in it. 1, 2, 3, not many more. And the carpels were infused. So these two-type structures here that you see in Rhabdopsis, they would have been split in the original carpel. It didn't have a transmitting tract because there's just 1 or 2 ovules. They will be met anyways. So much simpler. We know this not because we have worked on it, but someone called Peter Endres and James Doyle. They are like, well, the godfathers of comparative plant morphology, really. And you can do ancestral state reconstructions based on the anagrade, like the early branching angiosperms, and look how their carpels look like. And then you can infer from what you know from recent species on how the ancestor of this organ may have looked like. It's all inference, right? So there's nothing known that, in the fossil record that really shows us a carpel that looks just like that. So this is how we really can envision it. It's a simple thing. It contains an ovule and an ovary. The stigma is sitting straight on top of the ovary, and there's a little channel going through which is mucilage filled. As simple as that, most likely. And it's a big step from here to there. So this thing here requires 86 transcription factors, but it's a structure that you don't even have here. Right? So when we look at the ancestral carpel and want to understand how gene regulatory networks originated to form this structure here, and we have all the genetic information from that structure, it's going to be really difficult and more simple. So today, I want to talk about 3 questions. How old are gene regulatory network components? And are they angiosperm specific? Did all these genes that you need for making a Rhabdopsis carpel arise only at the most recent common ancestor of angiosperms or not? And then we want to learn about how to identify genes that are conserved in terms of sequence and expression throughout a large range of angiosperms, to really identify a core set of regulators that regulate carpel development in a broad range of angiosperms. And then I'll show you, hopefully quickly, an analysis that we have done on a non-model species using various induced gene silencing in non-Arabidopsis. Okay, so let's start with the first question. Many transcription factors are required to make an Arabidopsis carpel, and you have to simplify in some way. So what we've done is we've taken the Arabidopsis carpel here and just divided it into developmental modules. For example, there's a style stigma module, there's a meristem termination, organ identity module. So this is all published. And then we attributed some of the major transcription factors required for carpel development in the Arabidopsis to those modules here, just to keep it simple. Yeah, and you see, maybe not from the very back, but you see that some of the genes here, they turn up in several different modules. For example, an auxin response factor called etin is required for the style stigma module, it's required for the apical basal module. It's basically turning up everywhere. And when you knock it out, you get a really profound phenotype from a single knockout mutant. Okay, so we have these modules. And then my PhD student Kai did phylogeny analysis. We are quite good at this. And so he found out that these transcription factors are from very different families. And so he carried out phylogeny reconstructions for all these families, incorporating those transcription factors. So the rationale behind those phylogenies really was that you take the next closest gene to one of those transcription factors which has a very different function somewhere else, and this is your outgroup. So everything that's in the in-group would be homologous to this carpel developmental gene. And then he calculated and accumulated data sets and found that there are very old genes and there are very young genes. And I'll show you two examples. One very old gene. This is a gene from the group, the stylish SRS ones. When you knock them out, singly or individually, the style will not develop properly. So the pollen landing area is disturbed. And they have to do with auxin. So auxin homeostasis, auxin signaling is all funneled through those genes. What you can see clearly is that you have the outgroup down here, closely related genes Arabidopsis that have different functions, and the in-group contains bryophyte sequences. So this is an old gene. And then you have another gene, which is a young gene, the zinc finger transcription factor, which when you knock it out, the transmitting tract in Arabidopsis is not formed. And mind you, this is a structure that came only very late in evolution. And not very surprisingly, when you look at the in-group and out-group and which kind of species contribute genes to the in-group, you find that these things here, they are angiosperm specific, very young. So with this approach, we really can discriminate between old genes and young genes. And so we plotted those genes to our module, and got this. And what we found in short is that really all genes that are required for carpel development in Arabidopsis, they accumulated over time. So in every evolutionary step in land plant reproduction, you find novel genes coming into the network, right? So here on this side, on the left side, you see the state of the art, like all the genes that were present already in the most recent common ancestor of all land plants. Here. And you can see some of the transcription factors like the stylish genes that I showed before, Lunik and Sus, they were all already there. And then came some others, for example, auxin response factors, and so on and so on. And you can really see a steady increase. Yeah. So there is no such thing as the gene regulatory network of carpel is angiosperm specific. It's nowhere near that. There was no saltational evolution happening here, but it's a gradual increase of complexity in the gene regulatory network. We don't even know if those genes that are already present, say, in the most recent common ancestor of seed plants, if they were rewired in a network, because we can't work functionally in seed plants. Yeah? Because we lack genetic model plants in gymnosperms. This is really our blind eye. We can't work genetically with gymnosperms. And we're really sad about this. So, not angiosperm specific accumulated during time, and we know that they're rewired all nicely in Arabidopsis, but we can't really infer anything about their rewiring state and network composition in earlier land plants because we simply have only information of presence or absence of genes, not more. But then, we thought this is a bit of a limited approach, and we don't want to work in a Arabidopsis-centered way because you always have this bias, right? You find something exciting in Arabidopsis and you want to see if it's also conserved in other species. But Arabidopsis is a very derived species. The Brassicaceae are all out there, very specific. So it is much more likely that processes like development or general or more specific processes that occur in development are not very much related to what we see in Arabidopsis. And you can look, for example, in the at the morphological and also molecular genetics gap between monocots and Arabidopsis. When you, for example, just consider rice flower genetics and compare this to Arabidopsis flower genetics, you can see very limited amount of conservation there. So starting from Arabidopsis is not always the best way, because every plant species is successful because of their own ways, to adapt to the environment. And Arabidopsis is just practical, but not representative. So that's why we started a candidate-free approach. And we did this to really understand which genes are conserved between species of angiosperms, and which genes also show the same expression, like are carpel specific or are specific to certain time points in carpel development. And to do this, we used 4 different species, of course Arabidopsis thaliana, and then tomato, rice down here, and California poppy. So I showed you already the flower of California poppy previously. This is this butta here, and it's a member of the Ranunculales family, the buttercup family, which is really amazingly diverse, chemically and also in terms of flower development and reproductive syndromes. So we chose that because the Ranunculales are the sister group to all core eudicots. So before the asteroids and the rosettes, for example, split, the Ranunculales had already split off. And this is why we think that the Ranunculales may serve as a bridge to that. It's not working anymore, right? Still green. Did they do something wrong? What? Maybe the battery went. Okay. All good now. So we think that the Ranunculales species really bridge the morphological and genetic gap between monocots and dicots. So when we look at them, we find some genes that behave more like dicots and some other genes behave more like monocots. Okay, so those are the ones that we can also work with genetically. We can knock out genes in poppy, we can knock out genes in tomato and in rice, and of course in Arabidopsis. Now, PhD student here, he developed a technique called laser microdissection. Some of you may have heard of this. So we were actually keen to really have comparable stages of carpel development in those 4 species that I just talked about. And because the carpel is at inception really small and also in the middle of the flower, it's hard to get it. And then when you want to do gene network analysis, you need RNA. You need to know which gene is expressed when. And if you then want to follow gene expression over development, you need certain specific time points in development which are similar in all 4 species, and you need to be able to get to those 4 developmental time points. And this is why laser microdissection is such a good method, because it's rather simple. Well, it's easily explained, but it's really complicated and very work intensive. So what you do is you take material from, say, a bud, you embed it in a way that your RNA is fixed, you cryosection it, and then you put it on a microscope slide. And the microscope you use is one that's equipped with a laser, right? And so this is all done digitally, so you just have the section on your screen, you can label which part of the section you want to cut out, and then the laser does exactly what you told him, cuts out the section. And then you blow this section with an internal like a blowing mechanism into a little epi, and you collect many sections. This is the sad thing about many sections. And you can extract RNA from those and sequence the RNA, so do transcriptome analysis. So this sounds really easy, but in fact it's very difficult because the fixation mechanism to keep RNA intact is not really optimized towards tissue integrity. So you have to always make sure that you see the tissue properly that you want to dissect. And this is very challenging. Even more so when you have like so many tissue fragments that you require in order to have a transcriptome done from them. And then, before Kimo started his work, a few other groups had also used this technique on plants, so it's not nothing novel that we have done. But as we were working with four different species and only one PhD student, he developed a protocol for all 4 species. It's not a species-specific one, but it's a protocol that should work for a larger range of species. It's previously, everyone developed his own protocol. That was very laborious. So you can see a bit here how it looks like. So this is Arabidopsis Giessen development stage 1, 2, 3, and 4 here. For rice, the Giessen development is so simple that we couldn't find 4 stages because they need to be discriminated in a morphological way. So for rice, we have only 3. So this is basically the workflow here. So you harvest, you pre-process, fix, and cryo-protect. You do embedding and then sectioning and RNA extraction, and then send it out to the sequencing companies. And you can do some analysis with the results. So we started, of course, with Arabidopsis thaliana, just to see if our method is worth anything. It compares with what we know from Arabidopsis thaliana. And so what you see here is a GO enrichment analysis and clusters of co-expressed genes. So these are our clusters. For example, you have the 4 carpel developmental stages as 1, 2, 3, and 4 on the x-axis here, and we compare them with shoot apical meristem and leaf tissue derived from already submitted samples to the which we found in the NCBI server. So here in the top 2, we compare 6 different tissues, and then in the bottom 2, we only compare the 4 tissues that we have sampled, 4 different stages. So they are color-coded. So the blue ones here are strongly expressed in leaf and shoot apical meristem, but expressed at a very low level in carpels. And you can see that this corresponds, for example, to an overrepresentation of photosynthetic genes, which you would consider to be like that, because these guys here, the leaf and the shoot apical meristem, they are photosynthetically active. In these here, in the light blue ones, genes that are expressed in the leaf and shoot are expressed at the low level, but the 4 carpel developmental stages are really strongly expressed. And in those genes, in those clusters here, you see that photosynthesis annotated genes are heavily underrepresented. But what is new really is that in late carpel stages here, photosynthesis genes are again turned off in a very strong way. So the gynecium really, before the flower opens, is already photosynthetically active and produces its own carbohydrates. So it's not so much a sink anymore, but it's turning into a source. And when the flower then finally opens, photosynthesis really kicks in. So much of the carbohydrates that are sort of used up for producing the seeds are produced by the gynecium itself in the fruit. So we thought this was pretty cool. And lots of regulation going on, hormonal things, cell cycle, and so on and so on. So really cool data. You can also find de novo mutations with this approach, because we are looking for co-expressed genes, and some of the genes that are expressed in an interesting way, you can order rhabdosis mutants and phenotype them. And so we did find 4 mutants with a shorter fruit, and they are just waiting for hands to analyze those. Well, some further, because this is just the first step. Now what you see here is an overlay of the transcription data that we got with publicly available protein interaction data. So when people publish a paper on, say, protein interactions like yeast to hybrid or YFC or co-IP, and they are nice, they put the data also in a protein interaction network database. So you can extract from this database protein interactions that are experimentally proven, not somehow predicted, because you can't predict them properly. And when you then overlay this, you see something really interesting. This here are the protein interactions. Well, in the back it's really difficult to see. So these are little networks here of protein interactions. And then you see different color codes. And the blue boxes here, they are unique to that particular stage. For example, here you have a gene that's called CALCUTTA. This is the gene required for early Giessen development and is turned off after stage 1. So it doesn't show up in the late stage. And then you can really follow the transcription factor dynamics over time. Like there are transcription factors that are undergoing protein interactions with many other proteins, the so-called hub proteins. Here's one called topless. Topless turns every other protein into a repressor automatically. Now topless completely exchanges its protein interaction partners during carpel development. So the interaction partners of stage 1 are not maintained at all when you compare it with the late stage. It means, topless is apparently expressed throughout the carpel development, but it turns different genes into repressors while carpel development commences. So there's a shift of function of several of the proteins that we find here. And over here you can see the MADS network, the MADS transcription network. Some of you may be aware that those are important regulators, and they also change completely. So combining data, sort of doing a metadata analysis is a really interesting kind of approach that everyone can use because the data are available for free. Oops. And then, of course, you can plot it. You can plot it to the modules that we know from the last chapter that I talked about. And you can see that several modules that have genes that are old, occurring early in development, and other modules that were invented later in evolution with, genes that occurred later in evolution, they are present in the late networks. What this means, and we still need to really look at this some more, is that the early stages of carpel development may be higher conserved than the later stages of carpel development. So you basically put things on top of early development. It's getting more and more complicated, but these complications are not found in early carpel development and also not in early carpels, like the more original carpels. But this is just sort of a coincidence for now. We need more data to really confirm this. And then we have done some preliminary analysis also also with California poppy along the same lines here, except that there's no data set available for protein-protein interaction in California poppy, because there are only two handful of groups working on this, not like thousands like in Arabidopsis. So for protein-protein interactions, we have to do all the data by ourselves. So we have done a large protein interaction screen in the lab, and this is the large interaction screen is basically the work of Anna, which you haven't seen on the slides so far. And you can do, of course, heat maps of all the transcription factors that you feed into your protein interaction data set. And when we did it the very same way then in Arabidopsis, we were really shocked. Shocked in two ways, because expression pattern seems to be not so clearly defined in poppy as in Arabidopsis. It's lots blurrier. So genes are not turned on and off at a particular stage, but they are sort of, doing this. And no sharp borders like in Arabidopsis. So development in poppy seems to be more flexible. I mean, a bit more like Indian traffic maybe. So you sort of, you start with many different ways of doing things and eventually you get there. But there's no strict, you have just bus and car and bike, sort of the German way. And then the second aspect was that not only gene expression pattern, but also protein interaction patterns are much more promiscuous. These proteins, they really, almost every protein interacts also with another protein. Here. Bummer. We don't get networks, we get a big circle. And then we had to fiddle around with the resolution some more. And only consider very strong interactions and only consider very, very different expression patterns. But in a much more stringent way than in Arabidopsis. And you can see here we have much less data for Arabidopsis because we're doing it all for ourselves. But you see a similar pattern, also exchange of transcription factors over time. And we can also do some other more sophisticated analysis, bioinformatics analysis, like this one here, WGCNA analysis. This is what Dodo did. And we do find some components of the networks here, also in these co-expression networks. But this is very preliminary. It's preliminary not because our data are bad, but because they are based on an old genome. And now, just 2 weeks ago, the California poppy genome was basically released to us as a reference genome. And now we can much better match our transcriptomes to the new genome. Because this is, well, you all are probably aware that there are genomes published that should not have been published because their quality is so poor, you can't really work with them, and people got papers for not a good service to the community. So the new poppy genome will be really nice. We know because we've worked with this and we made sure that the quality is okay, and this will change the annotation and the read counts for our transcriptome substantially. So this is all very preliminary data. But this is also the very first analysis we have done. Meaning that those protein interactions we see here, they are pretty unique because you don't find them outside of Arabidopsis. The rice community doesn't maintain a protein interaction server and the tomato community doesn't either. So we can't do so many protein interaction analysis. So we can then only compare California poppy and Arabidopsis. But and now we come to the third, I'm not sure what time is it. Probably running late. Unlimited. Don't say that. That's too dangerous, really, because I can't talk unlimited. Well, I tried to wrap it up in like 10 minutes. So, Kai here, you know him from the phylogenic reconstructions, he found out that many of the genes that we looked at most recent common ancestors of angiosperms, they have duplicated in the core eudicots. So you may be aware that there's a core eudicot specific genome triplication. And many of those genes that were triplicated were maintained. They are overrepresented there. So most genes are being purged from the genome after genome duplication. But those guys, they were maintained. And so it happened quite often among those duplicated copies that one of the copies maintained its function in carpel development, one way or the other, but the other copy was involved in the Giessen zone formation. Yeah, because the Arabidopsis fruit dehisces sort of explosively at sort of breaking points, specific breaking points, and those are established by really intricate patterning of lignin production. And so it's complicated, and many of those genes here do contribute to this. And so we thought we'd look at this a bit further. And this is how the system really works when you break it down to a small set of genes only. So this is how Arabidopsis looks like when it's about to break. So two set breaking points. You have the valves here in green and the reticulum that remains when the valves disintegrate. And in pink here, you see the lignification, a row of lignified cells that curl up in a way when they dehydrate. And so this produces so much tension that during dehydration of the fruit, it splits open. Now this patterning process here requires a discrimination, a genetic discrimination of reticulum tissue and valve tissue and the Giessen zone, where the lignification will happen. And this is being realized by a small set of genes. Fruitful, shatterproof 1 and 2, reticulumless, indigissens and alcatraz. And all these genes, they all show up here in our modules. And when we did differential expression analysis in California poppy now, to see if the dehiscence zone is, or if these dehiscence zone genes are conserved throughout evolution, like that all the dry dehiscent fruits form their dehiscence zone in the very same way, we found this is not the case. Because some of those genes, I'll go back, like this one here, all those that are required to specify the lignification cells at the valve margin, they are core eudicot specific. So they cannot be responsible also for the lignification pattern in California poppy. So how do they really open up? And then another brave PhD student, Dominic here, who also likes to pick up people from airports a lot. He did a differential expression analysis of transcriptose collected from the dehiscent zone of California poppy to compare them with the non-dehiscent part of the fruit in early and later stages, just at the time when lignification occurs, to see which genes may be able to regulate the dehiscent zone formation. So this is how the Arabidopsis fruit looks like in a horizontal section. So it will open here at this W. There's a W here, there's a W here. There are two valves, one there and one there. And a little reticulum-like structure that will remain on the flower, or on the dried up flower. Now Dominic hand dissected those dehiscent zones at particular stages, and we look at the expression. We always do 3 triplicate and do those transcriptome analysis in triplicate, so you can be a bit more sure that what you see is real. And with this heat map, you can see how well those genes really are differentially expressed. And this is a set of genes that we know maybe, we sort of had informed hints that they could be responsible for the Giessen zone formation. And so when you follow the lines, there is the flow homeotic gene, C function gene, agamous here. There's another agamous here, and they don't, well, they do show some differential expression, but not as strong as, for example, something like this here. Seed stick. It's a gene I'm going to talk about further, because Dominic here, he has knocked it out. So, in Arabidopsis, seed stick regulates fruit length and ovule development, and also seed dehiscence. So it's somehow involved in patterning lignification, but not in the fruit, but it just ensures that the seed is able to leave the retinol. So this very small pedicel, this little stalk that connects the seed to the retinol, that the connection is lost, seed stick is responsible for. And the mutants, they look like this here. So the seeds are malformed and the connection is not being cut. So the funiculus I was talking about. So fruit elongation in this particular trait is is affected by the seed stick mutation Arabidopsis. Oh! No! Here. It hops. Okay, so we use a technique called virus-induced gene silencing for downregulation. And I don't really want to go into detail with this one, we can discuss this later. It is just a method that utilizes the RNA interference mechanism of plants targeted against RNA viruses, like plant RNA viruses. We meddle with the viruses, and then the RNA silencing mechanism that would usually silence an endogenous virus also silences an endogenous gene, our target gene. In this case, it was seed stick. And so Dominic did quite a bit of infection, looked at different plants, and we see that we can categorize the downregulation of gene expression into strong, weak, and almost none. So these here, they go down to 10% of remaining expression. So it works pretty well. And then you can look at the fruits and you see that, oh yes, there's some phenotype. They are smaller. They don't grow to full length. They dry out earlier. And they rupture open for some reason. So apparently the dehiscent zone is not, or the whole fruit is not strong enough to maintain tension until the very end. This is a bit tricky. Yeah. So this is how California poppy opens up normally. So these are untreated plants. So California poppy sort of dehisces explosively in a way that it like completely ruptures from the flower and sort of flows away, flies away. And when you then do the same experiment targeting GFP, this is our negative control because there's no GFP in the plant, so this is our negative control. It opens up the same way. But the seed stick plants, seed stick weeks treated plants, many of those, like up to 20% of the flowers of the fruits open prematurely. They maintain a small size and show this phenotype here. And then you can do histology. And up here is the control plant, our negative control, GFP VIX. And you can see that the overall architecture hasn't really changed all that much outside. but that this is the discrimination between reticulum and the valve margins is not as strong. So there's usually a file of cells here and there that is really thin and not lignified. And this file of cells also occurs in the seed stick plants, but it's much fatter. And what's probably more important for this premature rupture is that the inner epidermis of this seed, of the fruit, just continues to divide here. And this produces so much tissue, so much tension that the fruit ruptures very early. There are also some problems with the seed coat. So the cells outside of the seed, that are attached to the seed coat, they also grow abnormally. So apparently seed stick is required for restricting growth in the fruit and also for some patterning effects of lignification here and in the reticulum. So here you can see it a bit better. Wild type non-treated plants on this side, and you see this half moon structure here that is connected only weakly to the big fat lignified blobs here and by a file of unlignified cells. And this is completely ruptured, and this part here, the half moon shaped structure, extends much more. And this file of unlignified cells is missing. So there's definitely a patterning defect going on, but it's not so clear as we would have thought. So it's probably a combination of growth, restriction, and some involvement in the lignification patterning process that goes wrong when you take out seed stick out of the system. So, concluding here. If we come back to the three questions that I tried to address in the beginning, we can now say that the selection of carpel developmental regulators that we have analyzed shows that carpel developmental regulators were accumulated over land plant evolution. So there is no angiosperm specific explosion of transcription factors somewhere, somehow, that now contributes to carpel development. Can we identify common carpel co-expression networks in angiosperms? Yes. We still try, but it's hard because we just got the new genome and we have to recalculate all our data. So what I've shown you is that it's possible, but we need to fiddle with the data way more than we have done. And can we use non-model species to learn about non-Arabidopsis gene regulatory networks? Yes. With California poppy we can, because we have this beautiful tool by reduced gene silencing. We can also transform California poppy, but it's extremely laborious and doesn't work all the time. It's a difficult species. So, I have probably shown you some data to show that seed stick is involved in the hissensome patterning in some way, and also in regulating the inner epidermis of fruit growth, restricting extra growth. So, with that, I'd like to thank you for your attention, show you a little piece from my garden with California poppy growing here, and I'm very happy to take questions. Thank you.
Thank you, Professor Becker, for a mesmerizing lecture. I was taking notes all the time because I teach a bit of developmental genetics to them, so I know what to add now to my lectures. Thank you very much. So with this, as she's saying, she's happy to take questions. So can we have a few questions? Students of
Yes, semester, specifically student questions would be really nice.
Anything. So why are you interested just in California poppy? So focused on California poppy?
Yeah, that's a good question. Why are we focused on California poppy? So we started to work with California poppy simply because science and plant evolution to find a genetically tractable model organism.
Right, but
And outside California poppy, there is no such thing as a genetically tractable model organism. We would love to work with Amorella trichopoda, which is the sister species to all other angiosperms, but we can't even get it to grow. So we got seeds and they die in our hands. We go to Atlanta Botanical Garden, collect some specimens, and oh, the flowers all turned into male flowers. So they do crazy things. And it's for molecular genetics, it's really important. When you want to understand mechanisms, it's so important that you have a system that you can genetically manipulate. And they are very rare outside the coriodaikots and outside the grasses. And this is why we ended up with this.
Any other questions? Yes, there's a question.
Yeah.
I would like to ask you that what is the difference between the carpel of the California population is just originated in our bio and the carpel of the California population has just evolved and reached the latest.
Okay.
She's asking the difference if I can just
Yeah.
So you are asking about the ancestral form and the form that we find now. You are asking about the difference in the character. Probably she's asking about the ancestral coriodaikot carpel and the one that is now derived in the California poppy. What would be the difference?
Okay, so California poppy is not a coriodaikot, but it's outside the coriodaikot. It's a dicot, but not a coriodaikot. It sort of diverged earlier from the stem lineage of dicots. And so we don't really know much about the carpel of coriodaikots, the original carpel of coriodaikots. Simply because at that point, it's basically a whole genome triplication that gave rise or that sort of paved the way to the origin of all these coriodaikot plant lineages that we know. And there are so many that it's really difficult to do ancestral state reconstructions and reconstruct the coriodaikot carpel. But the difference of California poppy carpel to the ancestral carpel that I showed that was reconstructed is the same than the one in Rabbidopsis. Right, so they also have like a congenital fusion on top. There are two carpels fused in the same way that Rabbidopsis. The only thing that's missing is that it doesn't have a false septum. But everything else in terms of structure is rather similar in California poppy and Rabbidopsis. That was one of the reasons why we also used it. So it's much simpler than that from, say, opium poppy, where you have like hundreds of carpels fused together. Did that answer your question? Thanks.
Anything else? Any other thing that you want? Yeah, please.
when you said seed little bit food growth.
Yeah.
So is it inherent with plant growth hormones?
There are always connections to growth hormones. So if you just dig deep enough, you'll always find a connection of growth in cytokinin and growth in auxin. So I think there are no data so far of target gene analysis of seed stick, as far as I know. But I'm pretty sure there are some auxin response factors among them if you look for them.
Yeah.
Describe of the expression results that the so that is observed
early stage, we have talked about the carpels. So what are the monocots? Are there something conservative
Yeah, so we are at the moment in the middle of the analysis. We are actually waiting for a grant to be funded to analyze, to answer exactly this question. So the question was, so we have been looking at gene expression similarities between two dicots. What if we look at the monocot? Are there also conserved expression features that we can find? And so in order to do this, you first need to do sort of a morphology assessment to really see that you compare the proper genes with each other. And the further you go away in terms of phylogenetic distance, the more complicated this gets. Because the monocot lineage leading to rice underwent also independent genome duplications independent, and the core eudicots did the same thing. So the problem now is that we cannot attribute one-in-one ontology relations, because you may have 3 genes here and 5 genes there, and one of the groups, so these two groups somehow are homologous to each other. And then things get really complicated, because you're comparing expression patterns of groups of genes with expression patterns of other groups of genes. And we haven't done that yet, not with rice. Because it's demanding, so I would need a PhD. So I'm waiting basically for PhD students to start on that.
You have already spoken the habit of this. You have worked with that.
Yeah.
You have already spoken the habit. So what your support in habit is not justifying or satisfying your kind of work with other type of endocrine type.
Yeah.
So can you so for monocots, can we work with only rice just like habit of this? Can they justify the work like habit of you already spoken the habit of this? It's not justifying the work with other kinds of diets.
Yeah. So the problem is that we are lacking model plants in the monocots outside the grasses. So it would be so nice if somebody of you maybe would come up with, say, banana or an orchid. So orchids are transformable by now. But this is basically in the hands of very rich orchid breeder companies, and they do not share much. That's a problem really. But it would be a wonderful model system to test these genes also in orchid, but we don't get our hands on orchid transformation. And so the morphological differences in Giessen development between the different grasses is not so great. So we would need species outside the grasses genetically tractable, and I can't see this at the moment. So I don't think it's worth looking at 5 grasses, because they will be all very similar. But banana, I'd go for banana.
For viruses gene transfer.
There are viruses for banana. But then you have the generation time, and well, you can't grow them in large numbers. Also quite nice. Yeah. For one of those viruses, gene science experiments, we need like 150 plants. And that sort of is a lot for bananas.
Most of the banana in the relative family.
Yeah. They're just pretty small bananas.
She is working with gingivitis.
I was too excited genetics, not She works in ginger, that's why I'm scared of bananas.
Yeah.
Is there any mechanism for the in-dehiscent fruits? For example, in case of the dehiscent fruits, there is a particular layer and some genes are also present. Those fruits which are indehiscent in nature, is there any such a which one is considered as the primitive?
Primitive is indehiscent. Indehiscent is.
So the development of certain genes is responsible for the dehiscence.
Well, we by now think that dry dehiscent evolved several times independently. specific with different gene sets involved. Because the original fruit didn't have any DNA, so it was probably just sitting somewhere in the ground waiting to decay, and then the seeds would germinate. Nothing specific about that one, yeah. So multiple independent evolutionary sources.
So that's a relative question. So for seed state, for those plants where they don't have this kind of state of the mechanism for seed, any sense like so what is the status of functional seed state in those, you know?
Well, in seeds again, say tomato. This is like a classical non-delisted fruit. Seed stick is still present, and it's required for funiculus development and also for fruit length. Yeah. So that it is involved in fruit dehiscent in California poppy is probably new functionalization that was probably somewhere within the ranunculales. So we haven't even looked at other ranunculales. So in our system, silencing, so depends. So the question was how many genes can be silencing in our system? So we need around a fragment of 350 base pairs. And then when you have genes that are closely related, you can choose these 450 bases wisely. That you target a conserved region, and you may be able to target several at the same time. But then you still need to do the analysis. You need to do qPCR to see how successful you were. Most of the time, you don't get them downregulated all at the same level. So we've done 2 at the time, other people have done 3 at the time, but the smaller the fragment gets, the less efficient the downregulation. And you can't, so these viruses, when they are active in a plant, they still produce a capsid. And so the amount of DNA that you put into the virus needs to fit into the capsid. And so rod-like viruses are the best really for those systems because they just can extend the rod length. But many viruses are not rod-shaped, and then you have sort of a virus stability problem. It all depends on the virus. There's viruses that can take much more, but you use tobacco-virus, and it takes maybe 700 bases extra load, but not much more.
Maximum percentage of downregulation?
Well, we can downregulate up to 90%, but this is a really messy system. And so if we have an experiment that runs really well, the range is basically from 90% downregulation to 10% downregulation. Some plants, they are so happy living with the virus and they don't show anything. No. Well, shoot apex. We infiltrate the shoot apex with a very thin syringe. No, we infiltrate transgenic agrobacteria that carry those viruses as vectors.
Yeah, there's a hand up, please.
Yeah, okay, there are many ways of gene silencing. So you can do microRNA silencing, or you can use artificial microRNAs, and you can use other siRNAs. You can do, of course, CRISPRings, all very nice, but for all these mechanisms, you need a transformation and regeneration protocol. And this is really the key. This is the main problem that the plant community, the plant evolutionary developmental community has, that there are only very few model systems which you can transform stably and regenerate. So we can do this in California poppy, but it takes a year with lots of work. So we choose virus loose gene silencing for now as sort of a dirty way of getting first ideas on which genes to look at. We are currently working at the CRISPR protocol, but this is still a way to go. So virus loose gene silencing, the beauty is that this system works systemically. So all you need to transform only a few cells with agrobacteria that carry the virus on a vector. And the the plant's immune system will recognize the virus, start the RNAi process, and then spread systemically throughout the entire plant. So when we, for example, we do the experiment with a plant that has only 3 leaves and is like 5 weeks away from flowering. But we can analyze flowering phenotypes and we can analyze fruit phenotypes using this mechanism without the need for stable transformation.
Yes. That's a minus. We are doing it.
So, if we, yeah, there's another question. Yeah, please.
Which one?
Yeah.
Left one. This is the morphology of the California poppy fruit when you do a section, a horizontal section. Oh, okay. This is the log 2 of the reads per million. So it's basically a measurement for differential gene expression. Yeah. So the bigger it gets, the less strongly it's the more strongly expressed the gene is, and the bluer it is, the less strongly expressed the gene is.
I am talking about that problem. So in the left hand side, the upper portion.
The section.
She is asking about this one.
Ah, okay. Oh. Okay. This here, section of California poppy fruit. So when you slice through it horizontally, you see it looks like this. So you have here 3 seeds, and these blocks here, they are fat lignified ridges that keep the California poppy fruit upright. So the fruit is about this long when it's mature. And so these 3 here are for stability, and the dehiscence would be here. Here's the dehiscence. So you have one carpel here, one carpel at the bottom. Here's the rectum, and here would be the dehiscence.
Okay, so does that answer most of the queries that you guys had? You have another question? Yes? Follow? Yeah.
You are already you mainly want California. I think it's mainly California. So what kind of work can be done in children?
Yeah. So, well, everybody can grow Arabidopsis, can grow California.
She did not choose the plant because it grows in a particular
Yeah.
for its evolutionary importance and its position in the phylogenetic tree of the animal.
And its practicability.
And its practicability. So if you can find a plant like that in India, you can go.
Yeah. If you find an interesting species that you hope will answer your question, you can also turn this into more species. I know an Indian postdoc working in Germany at the moment. He works with an amphibic plant that shows a very strong case of heterophily. So I've been in contact with him a bit because the leaves of this plant look very different when they are submerged versus when they are growing out of the water. And this is a perfect model system. I mean, unlike we suffer with poppy quite a bit. But this plant has a small genome, transcriptomes he has already, and he can transform it. So within like 3 years, he has turned an Indian species into a genetic model system. Very impressive, very smart.
You have just explained the new cutting technique under microscope,
Yeah.
analyzing it under the microscope. So can we just transform a microscope into that cutting machinery like existing microscope, or is it a new system developed?
It's a new system really. Yeah, because you need to equip a standard type microscope with a laser that's powerful enough to section out certain parts of the slide. And then you need to also come up with sort of a blowing machine because you can't touch the fragments that you get out. And you need to transport them into an equitable connection. And so the laser needs to be very close to the lenses. And I think this is challenging. So this will be difficult to build, I think. Very sophisticated.
Do you ever have this problem of not having sufficient RNA out of fish?
Oh, we always have this problem of not having
And we
sufficient
had to,
RNA.
and once you use this artificial RNA amplification method to increase the quantity of RNA many years ago, but then that adds this artificial free-time virus and all these things. How do you deal with this? The tissue pooling is going the only way.
Well, there is, first of all, you need to do lots of tissue pooling. And it's not just a problem of RNA quantity, but also of RNA quality. So degradation really is a massive issue here. So the sectioning is all done at -20 centimeters. And we have a particular kit for low input RNA extraction, and the sequencing protocol is optimized to low RNA sequencing libraries, and there is a single amplification step involved. A large problem here is that when you do normal transcriptomes, you can select for non-ribosomal. You can somehow remove the ribosomal fragment. And this is very difficult for small amounts of RNA. This is really challenging, that when you have those reads, 50-60% will be rRNA. So you need to sequence more. Yeah. And the question, of course, now is, well, we are studying at the moment how the RNA integrity values really affect the sequencing outcome. But this is an ongoing project. So RNA extraction is always a problem.
Yeah.
Yeah.
As we all know that all the setting is made of because there are like my partner so I will also what is
Okay, so the question is basically, we are looking at transcriptomes and expression analysis, but there's so much else coming afterwards, right? So post-transcriptional regulation and, well, export of the RNAs and protein translations and things like that, we don't. Simply because we can't. Because the layer of complexity that we're looking at the moment is already so massive that we are overwhelmed. If we would now combine this even with proteome analysis, we'd go nuts. So we'd love to, but it's completely out of our scope at the moment. Maybe at one day when there are some better algorithms and easier ways to get proteomes from small amounts of tissues, this would be wonderful to have. Yeah. Because we know the gap is there, but we can't fill it. That's a very good question. Yeah.
So I don't think I have any questions. I just asked if they say that there were no questions in the chat box. I just took Matt's permission that you can ask her questions also about if you're planning a research career in Germany, or if you're going to apply or you're applying for a PhD in Germany. If you have some queries regarding that, or if you would want some advice regarding that, you can ask those questions also. So it's one hand up.
I would be very, very interested to know a bit about the application process, because I'm in the process of applying to different labs, particularly in Germany. So I have a bachelor's in Botany and a master's in genetics from the Justus-Liebig-University. Some research experience working with investigating transcription analysis using cell approach prognosis.
Okay, so the application processes are multiple basically. So if you want to do a PhD in Germany, first of all you need to know that there are no fees involved. So doing a PhD means you can do only a PhD for insurance reason when you bring a fellowship or a stipend, or when you have a position in Germany. When your parents are really rich and they want to send you for a PhD to Germany, we cannot accept you because you need to bring funding, like proper funding, not parent funding. And so that then relates to how to get a fellowship. So the IAD provides fellowships for PhD students from all over the world. It's quite competitive, and that involves that you, before you even start thinking about moving to Germany, you need to contact first a German professor. Without using the word respected sir, I would like to work with your esteemed research group. Skip that. They go straight into the trash, really. But if you write to someone saying, look, I would like to work with you for scientific reasons. I've read these papers and I really, really like what you do. And I went through your homepage and I see these are the projects that are running in your lab, and I would like to be part of this. And I bring in the lab, not just I'm very good at all sorts of things, but I have experiences with the following techniques that may be of use to you. And that does not mean excellent. And it does not mean I know how to drive a car. But that means, for example, I have done digital gene expression analysis, or I'm really good at cloning, or well, you name it. You see what we do. So hands-on experiences is what counts. And that does not include really what you have done in some practical courses. studies is what you have done independently in the lab at your own responsibility. What you can do, what you can contribute. And then you will become interesting to the PIs all over the world. It doesn't really count only for Germany. And then they will open up and provide you with opportunities. There may be fellows. So the DAAD is one way for fellowships. I think Humboldt doesn't provide any PhD fellowships.
It was 3 years ago I heard.
Okay. Yeah.
But I think somehow this is only some
Yeah, so I'm not sure who what does, but there are also other funding agencies in Germany. Usually the universities don't have, unlike some British and American universities, German universities don't have their own fellowships. That means that professors that employ PhD students have to write a grant application. And for these grant application-based positions, you need to be better than their students if you want to have one of those. So it's also worth looking at positions that are announced in Germany and see which ones you would match to. And when there's nothing around that matches your expertise and your interest, you do this writing that I just explained. Very specific, project specific, so the professor knows that he or she, and only he or she is meant, and not 1,000 other professors in Germany. And then they will help you with the application, because you do not only need a letter of, like a statement that they will keep you in the lab and maintain you with things, but they will also for sure need to help you writing the application. So that your application would be integrated well into the lab, and that really improves the chances of then getting a fellowship. So, because these grant applications for, let's say, DAAD or other fellowships, they are very competitive. So just writing up something will not help. It has to be a very well thought through research program. This is difficult for someone at your stage. It's even difficult for me to write a grant application, and I've done like many. So very targeted. Yeah.
Almost association or Max Planck paper, do they have fellowship or it's only for the project?
I'm not really sure if the Max Planck and the Helmholtz associations have one, or the Fraunhofer. I know that the Max Planck Institutes, they provide their group leaders and PIs with more positions that they can choose for. There's also more pressure at Max Planck. Yeah, definitely. Because the group leaders, they are funded at the Max Planck for 5 years only. Not initially, 5 years, that's it. So after 5 years, they need to find their own position, meaning 5 years of massive pressure. And I mean, they are not in the lab, they are writing, so who's getting pressure then? It's the PhD student, the postdoc working with it, just under pressure. So yes, they are on very volatile positions. Very well equipped, wonderful environment, but there's pressure. So there is actually a website of the DAAD that lists fellowships additional to the DAAD fellowships, and they may be like subject specific or stage specific. So it's really worth looking at this website. It's very well done and very well maintained.
So I think we can bring an end to the question and answer session. If you don't have any further queries, and I would thank the Department of Botany, University of Calcutta, and the Sharma Foundation, and of course, Dr. Maura Raj from Residency University. We are a contact person that we had Professor Annette Becker here. And I request the Head Department of Botany and the President of Archana Sharma Foundation to give our small token of thanks to Professor Becker. Wow! This is wonderful.
Madam, just a second. Actually, yesterday we had a meeting with next plant Botany Institute, and she was carrying my daughter. They were carrying flowers to one market, and all the visitors were coming. They were gifting them one flower. Yeah. Because she was talking about flowers.
So this flowers, these flowers also have carpels.
Thank you so much.
Okay.
And I request the Head Department of Botany for the customary vote of thanks.
So, on behalf of my Department of Botany, University of Calcutta, and on behalf of ASFC and on my own behalf, I would like to thank you all, and especially I want to thank Professor Becker for accepting our invitation and delivering the illuminating talk. We were really mesmerized, and I hope her delivery also incited many questions among you, the young minds, and obviously you will try to solve some of the queries she has raised among you. And I would like to thank our Vice Chancellor for allowing us to hold this meeting, and at the same time I would like to thank Dr. Monoy Dash for bringing Professor Becker here, and I would like to thank all the students and participants for joining us today. Thank you very much.
So that ends the session here. There is some refreshment, coffee and something else outside. So please